So it is my pleasure today to introduce Professor Feng Peng from Wuhan University. She's a professor of microbiology, and she's part of the team that leads the China Center for Type Culture Collection. And she is very active in polar research, polar microbiology research. And today she's going to give us an overview of the work that she's been doing in the Arctic, which is something we don't often hear about in New Zealand.
Thanks very much. Thanks very much for your invitation, and thank you come here to hear my research. And it's my honor to share my research there. All right, let's start. My research in this presentation is about microbe activity under frozen condition. Okay. Yeah, I'm a microbiologist. Everybody knows the microbe is everywhere and play an important role in the region, in the polar region, especially the harsh environment, And so my research interested, for example, novel and special microbes and to their diversity, survival strategies, function, and evolution. I've been to Antarctic 4 times and to Arctic 9 times, so more times in Arctic. It's really a core research in cold region like that. Yeah, and also I have got some warmer friendship. So let's introduce our group, the cordial group. There are 5 teams from CAS and Chinese CAS and the universities. Cooperation to form the cordial group. The vegetation cordials were established for the long-term monitoring in Arctic and Antarctic. It's a platform for interdisciplinary research in botany, microbiology, ecology, geology, and meteorology. Yeah, this is a cordial. The cordial we need, of course, a different person will research on this one. So we need the higher plant in this cordial, and the lichen and the moss, and of course the soil. The sample for me. And this is the monitoring area, and this is the sampling area. The soil temperature, salinity, moisture, and irradiation will be monitored. And the ecological effect, response, and the feedback of the microbes and vegetation in the cordials, and also the aerosol or pollute in the cordials to the environment and the climate change will be studied. Well, in Arctic, my main sampling area in the New Zone of the Svalbard belong to the Norway. I was standing on the site at the edge of the glacier 3 years ago. So there are more than 30 meters away the current edge of the glacier. So the climate change in polar region is really evident. Since there is minimal impact from the external environment, so it's an ideal place to study the microbe succession. Well, we established 4 cordials in the retreat foreland of the glacier, the Australopithecus glacier. It's a small valley glacier. And glaciologist provides a retreat age of the glacier that helps us to establish the cordials along the glacial retreat coral sequence in different age. The Cordial 6, the Cordial 6, this week, well, the Cordial 6 looked at the edge of the glacier 30 years ago. Is particular early stages of the succession, and with the pioneer plants and a few mosses there. And Cordial 7 looked at the edge of the glacier 80 years ago, and with the significantly increased of the plant species. And the Cordial 8 located at the edge of the glaciers maybe about 200 years ago, represent the mature stages of the succession. And the plant coverage increased. And the location of the Cordial 9 didn't covered by the glacier in 2000 years during the Little Ice Age. So just as a reference, Cordials. This is my slide. And sorry. Yeah, we monitor these kinds of cordials. You will see that the French group help us to do like this. Then the samples collected from the cordials in spring and in summer. The DNA and the RNA were extracted from the soil samples. Actually, it's not easy. I think you know, because especially in the frozen soil, the RNA is the quality and the concentration of the extracted RNA is so less. So I had to just for the RNA sequencing. So through the high throughput DNA and the RNA sequencing, the diversity, composition, seasonal variation, and the succession, also the activity and the adaptability were studied in this in the foreland of the glacier. And we want to know who is there and who is working, who works hard, and how it changes with time and seasons. But this is the cordials. It is in summer. One of the cordials there, you will say it's really barren. Little plant, a pioneer plant in there. This is a village, small glacier. And also some equipment set here is for collect alcohol. And it's the with the got against the bluebell. Sometimes when we set in, the bluebell will wait us. So it's sometimes a danger. Well, in summer, simply it's okay, but in winter, well, of course, the windows also have a thin ice layer on the corner. We need to empty to put sampling. It's really not easy. Okay. And sometimes maybe we establish the cordials, but next year we back to this place, we found it's destroyed by reindeer. Of course, they have a reindeer. And also sometimes we met some dangers. This 2 years ago, I slipped on the glacier and broke my ankle, so I have to ask for the helicopter rescue. And also, the most danger is from the polar bear. This is morning, 2:00 a.m. Of course, it's active the polar day, so it's also sunshine. You will see the polar bear. It's the first time in summer the polar bear went to the station. This is in New Orleans. There are 8 stations from different countries. This is the German station. Of course, here outside of the house have reindeer skin, so we think the polar bear is really hungry. So what it is, I don't know what will do if you know the polar bear outside your house. You will feel, for us, will exciting. And go outside, take picture, review. And this video is we stand on the roof of the house and to take the video. And this is the sculpture of the Amundsen, the person first arrived at South Pole. He is the hero in Norway. So happy for him. So anyway, the sample is not easy, but we also get some result about it. I will show this. In our research, the DNA sequence indicated the total microbe community, and the RNA sequence indicated the potential active microbe community. According to the Chow and the Channel analysis, we found the richness and the diversity, along the retreat region, along the retreat coral sequence obtained through the RNA sequence are more regular in and have the same pattern in summer and the spring. That means the succession of potential active microbe community are more stable in different seasons than that of the total microbe community. For the bacterial community, we can see the richness and diversity increased along the chloral sequence. And for the fungi communities, it will be increased first and then decreased. But for the aqueous communities, the richness will be increased first and then decreased. But the diversity will be the diversity was increased in both seasons along the chloral sequence. Well, through the DNA sequence, yield more phyla, but not significant except the outro. And also, there are no significant difference between the DNA and the RNA sequence for the predominant phyla, but the content of the phyla it also have a seasonal difference. We can find some phyla have more content in spring, and others maybe in the more content in summer. Also, the succession along the chloral sequence is also have the seasonal difference. Some phyla increase in both spring and summer, and some just increased in spring or just in summer or decreased. But we also find for the DNA and RNA sequence, the pattern of the succession were the same through the RNA or DNA sequence. But also, some finally have the opposite pattern of the succession. For the PCO analysis, the potential active microbe communities of the different cultures were separated, but the difference between the DNA and RNA sequence is significant greater than that in spring and summer. This RNA sequencing and this DNA sequencing, so it's really different. For the perma-novel analysis, we found the succession of the potential active microbe communities related to the nitrogen, in both seasons related to nitrogen, the phosphorus and the sulfur. But in spring was most related to the nitrogen and the sulfur. In summer, most related to the phosphorus and the coverage of the plant and the species. Well, since the increase in the content of potential activity bacteria may be due to the increase in the numbers of the bacteria or cellular activity. So the 16S RNA ratio compare the RNA and the DNA content directly was used to assess the microbe activity. We will find some final have the high 16S RNA ratio, but also some final with the low 16S RNA ratio. Notable, the red one. The red one is the dominant phyla. So in our result, not all dominant phyla has higher 16S RNA ratio in this region. That's why we think maybe the RNA sequence reflect just the sampling time, the actual time of the sampling. So sometimes maybe some the phyla grows quickly, but the nutrient used up or the pH or change it, and their activity is not higher activity. Yeah, we think maybe for this is. Yeah, we know a lot of studies have shown a positive correlation between RNA ratio and growth rate in the pure culture. And then the 16S RNA ratio is considered to be an indicator of the microactivity in glacial communities. But that course, most of the time, the microbes so small, we evaluate their activity based on the growth and the production. But we know the microbe activity does not equal the growth and the reduction rate. So what is the relation between the changes in 16S RNA ratio and no growth activity? What kinds of metabolic activity? Anabolic or catabolic or energy metabolism? So for research, for example, since we select two genres based on the 16S RNA ratio rate with the opposite pattern. The Petobacter have a higher 16S RNA ratio in spring, and Bacilla have a higher 16S RNA ratio in summer. Then the two strains isolated from the cultures were incubated in the 10-fold dilute R to a medium, and at the minus 20 degree or 10 degrees, to mimicking the oligotrophic and low temperature environment in spring and in summer. And after 5 days, the 16 sRNA ratio and the CFU and also the CTC damping straightening that's tested the respiratory activity, and also the ADP were detected, were measured, and also the transcriptome and tabulum were detected. As a result, of course, in these two strains in a harsh environment, so the 16 sRNA ratio of the both strains decreased. But in Helicobacter, the 16 sRNA have a higher 16 sRNA ratio at minus 20 degree. Well, the Bacilla have a higher 16 sRNA ratio in at 10 degree. So it's consistent with the pattern that they in the spring and the summer in front of the glacier, right? And this result is also consistent with the mortality rate and CTC damping ratio. That means the Helicobacter have a higher survival rate and higher 16 sRNA ratio and the higher respiratory activity. Well, the Bacilla was opposite, but the ADP is different. Both the strain have a high ADP at minus 20 degree. That's very interesting. So we think it seems the higher 16 sRNA ratio indicated a strong adaptability. Then for the transcriptomic analysis, we find there are more upregulated genes in of the two strains, both at minus 20 degree and 10 degree. And at minus 20 degree, both the strain upregulate the translation and energy metabolism. The enrichment of the energy metabolism could account for the higher ATP concentration observed at the minus 20 just now. And also for Helicobacter at minus 20, upregulate the gene related to the amino acid metabolism. Actually, it's for the biosynthesis of the amino acid. But for the Bacilla, have more gene upregulate relate to the carbohydrate metabolism and membrane transport. And also, we find some cold shock protein exchanged at minus 20, but in Helicobacter have a significant more the content of the upregulate of this cold shock protein. Well, as we know, in response to the stress, for example, the starvation and the osmotic shock, the SPOT or RELA will transfer the ATP to GDP or GDP and synthesize the PPGPP together with the DSKA or the different sigma factors. binding the RNA polymerase and redirect the translation from the growth-related gene to the gene involved in the stress-resistant or survival. Then we find out for the transcriptome analysis, we find the RELA and the DKSA gene upregulated in macella. In macella, significantly upregulated, but SPOT gene down-regulated. Well, the Heterobacter was opposite. So we think maybe it's possible the frozen stress initiate the synthesis of the GPP and block the production of the rRNA. But the upregulated of the SPOT in Heterobacter some different. Of course, the SPOT is not just synthesis the PPGPP, also can degrade PPGPP, have a balance, depend on the condition of the cell. It's complex actually. All right, so in Heterobacter, SPOP synthesizes the PPGPP and then initiate the amino acid biosynthesis, but it also can degrade PPGPP. So the reduction of the RNA is not so significant in Heterobacter. We think maybe it can be explained. It can explain why the Heterobacter have a higher 16S RNA at -20. Also, for the metabolomic analysis, both the strain at -20 produce the short peptides, but in Heterobacter have more significant amount of the short peptides at 3 to 4 amino acid at -20 degree. That consistent with the transcriptome analysis. Of course, in the Heterobacter, the transcriptor gene of the analysis biosynthesis upregulated at -20 degree. So, summary, in our research, the diversity, potential activation, and the succession of microbe community along the great glacial retreat cloud sequences, spring and summer were explored. And we know who is there and who is working and who works hard. The interesting, we found some final with higher 16S RNA ritual in frozen spring than in melting summer. And then the two strains were selected for the no growth activity research. The result linking 16S RNA ritual to bacterial adaptability. For these strains, we find the frozen adaptation strategies, including upregulated energy, metabolism, translation, expression of the cold shock protein. For Heterobacter, the increasing amino acid synthesis and short peptide may lead to the higher or stronger adaptability. This association between 16SR ritual and environmental adaptability help us to screen the circle trophies. Of course, we don't know, before our research, we don't know the Heterobacter is psychotrophic. Actually, it is coarse. It couldn't grow at minus 20 degree, but it can grow at minus 4 degree. You'll find. And the research on the microbe activity shift across seasons and successional stages will aid the prediction of the ecological responses. I think our research might explain why the greenhouse gas emissions suddenly after the snow melting in this region and higher than in summer. Of course, under the frozen condition, they also can work. They also have the activity. Well, I'm a taxonomist. I like to isolate novel and special species and establish a Polo microbe resource bank. It's public open. If somebody have interesting, I can connect me. It's published open. If you have interest about the cyanobacteria, I have some different. Yeah, you can use that. Well, welcome to our center, China Center for the Culture Collection. Next year we'll have a new building. Welcome to visit our center. And also welcome to my beautiful university, the most beautiful university in China. Welcome to China. Okay, thanks my groups, my colleagues, my students, and thanks you for your attention, for your listening. Thanks a lot.
Really interesting talk. Are there any questions in the audience, from the audience, in person or online? I have a question. I'll start with a technical question. How did you stop R2A medium from freezing?
It's frozen, surely.
Yeah. How did you do that?
Yeah, you mean for the first sampling? Sampling should be very careful.
No, not sampling. When you did the experiment at minus 20, how did you stop the R2A medium from freezing?
Yeah, I will tell you.
Okay.
First, for the sampling, we will put the frozen soil inside the protected solution, protective soil solution, and when we back, it will melting in the solution, and then we will frozen it in the protective solution, frozen it and back to the laboratory, and in the laboratory we will melt it. Of course, it's in the protective solution, so we think it will stop the RNA. So it's working.
No, but when you do the experiment, right? When you were doing the 10 degrees, minus 20 degrees experiment, what did you do to stop the medium, the growth medium, from freezing?
Yes, I told you just now, the Petrobacter can grow at minus 4 degrees. Yeah, the solution, the medium solution is in the medium solution. It's frozen.
It's frozen. It was.
It's frozen.
So that's really interesting. So basically, and so my next question then, before I look at my next question, are there other questions? Joshua.
I have more of a naive question. So it seems like so ATP as a recording. It seems like that there's activity happening at both temperatures, and yes,
That's
and the
sorry, I think it's okay.
and that the temperature does serve as a differential of which microbes are active. So is this perhaps a way of reducing the competition at a very, very low class scale, based off of temporal differences of microbial activity?
Somebody used the method ATP to assess the activity of the microbes, but in our research and find in a frozen condition, both strains have a high concentration of the ATP, and especially, of course, the Messina is actually have a less survival in the frozen condition, but it have higher ATP concentration than Helicobacter. Okay, we think also we read recently a paper published in the Nature Committee on that. It also finds the similar things like the course one bacteria grows or reflect very quickly, grows very quickly, or activity, they will they will use many things. So the energy, the higher energy, it's not good for them survival actually. But in a cold place, like our person, if the cold, we need energy. Yeah. If I cold, if I wake, I need more energy like that. Okay.
So my other question, so I'm using the microphone for the benefit of the online people. Are there other questions before I ask my questions? Stephen?
So, so you mentioned that some of your species have a trend where they're more abundant the further along in the chronological sequence they are. Their DNA showed that trend, but the RNA showed the opposite trend. Do you have any ideas why?
I think maybe, yeah. Course at that time could have a balance of the numbers and the activity. Maybe for the DNA sequence, it increased for the numbers of the bacteria, microbes. But for the RNA, the cellular activity is not like that. Maybe in the like the Helicobacter in a frozen place, in a frozen condition, have a higher activity, but the number of the bacteria microbes is not higher. I think maybe like that. I have a question also.
My question is actually about the bit that you didn't talk about very much, which is your culture collection. So these are all Arctic and Antarctic isolates. Yes. Have you measured the optimum growth temperature of these ones?
You mean store the strains in the.
Yeah, the strains in your culture collection. Do they? I guess my question is do you see that they are actually growing the best at a low temperature, or are you seeing that they're quite happy at 30 degrees or?
Actually, it will be happy in higher degree because when we isolated thank you a lot. We isolate the strains from the low temperature, maybe 4 degrees, but we test the growth degree, we will find out maybe 20 degrees is good for them.
So that actually was my question. But the two species you looked at, what were their optimum growth temperatures?
20
So
degrees.
They're both cyclotrophs.
No, I think the Micilla is not a cyclotroph. Yeah.
Does it only grow?
No.
It's really interesting. It's not surprising, right?
No.
I think we assumed that another bit that I noticed that sort of echoes what we see in Antarctica as well is that people assume that there's a diurnal activity pattern because people measure them and they think it's the error pattern. But it turned out what they were measuring was the quirk of Antarctic soils, because Antarctic soils are very high in carbonates. And so when they do CO2 flux, all they were seeing was the carbonate coming in and out of solution driven by surface temperature. And so when we actually did a continuous carbon dioxide monitoring, we saw that nearly all of the biotic flux occurred in one peak, the first time the temperature got above freezing. That was it. That was all the pent-up energy they accumulated over the years. They just used it all up in one go, and then everything was really went back to baseline.
That's interesting.
I think the Arctic is very different geology, so I don't think the Arctic has nearly as much carbonate in the soil typically.
Yeah, we can compare the data about that.
Yeah.
I have no idea what is a higher, what is that.
Yeah.
Yeah.
Absolutely. I didn't see any.
I didn't see any either. I knew it popped up.
Well, Professor Payne has included her email on the slides, so I'm sure if there are questions, people can email Professor Payne